A video about atavism. I shouldn't say I'm a fan of Friedrich Nietzsche, but I am someone who has read the works of Friedrich Nietzsche. I used to own all of Friedrich Nietzsche's works, and I've read all of Friedrich Nietzsche's works. One of the things that Friedrich Nietzsche was very accurate about was his bashing of eugenics as eugenics was at the time, as he saw eugenics as a domestication of the human animal. So what is atavism? In biology, an atavism is a modification of a biological structure whereby an ancestral genetic trait appears after having been lost through evolutionary change in previous generations. Atavisms can occur in several ways, one of which is when genes for previously existing phenotypic features are preserved in DNA, and these become expressed through a mutation that either knocks out the dominant genes for the new traits or makes old traits dominate the new one. A number of traits can vary as a result of shortening of the fetal development of a trait, blah, blah, blah. So what I want to point out here is Ragnar Redbeard is another one that points this out. You know, what good is selective breeding if selective breeding results in a bunch of milk-sopped weaklings that chitter-chatter about Play-Doh and crap like that? That should not be the goal of things like eugenics, which, you know, eugenics should not be to domesticate and weaken the human animal and make a bunch of useless eggheads out of them. In the social sciences, atavism is the tendency of reversion. For example, people in the modern era reverting to ways of thinking and acting of a former time. The word atavism is derived from the Latin atavis, a great-great-great-grandfather, or one or more, generally an ancestor. Biology, evolutionary traits that have disappeared phenotypically do not necessarily disappear from the organism's DNA. And I'm going to bring this up with epigenetics in a bit. The gene sequence often remains but is inactive, such as an unused gene remain in the genome for many generations. As long as the gene remains intact, a fault in the genetic control suppressing the gene can lead to it being expressed again. Sometimes the expression of the dormant genes can be induced by artificial stimulation. Atavisms have been observed in humans, such as with infants born with vestigial tails. Atavism can also be seen in humans who possess large teeth, like those of other primates. In addition, a case of snake heart, the presence of coronary circulation and myocardial architecture that closely resemble those of the reptilian heart, has also been reported in medical literature. Atavism has also recently been induced in avian dinosaur bird fetuses to express the dormant ancestral non-avian dinosaur non-bird features, including teeth. Other examples of observed observed atavisms include hind limbs in cetaceans and sirenians. Cetaceans are dolphins and whales. I don't know what sirenians are. Extra toes of the modern horse, reappearance of limbs and limbless vertebrates, re-evolution of sexuality from parthenogenesis in oribetid mites, teeth in avian dinosaur birds, dew claws and dogs, reappearance of prothoracic wings in insects, reappearance of wings on wingless stick insects and leaf insects and earwigs, atavistic muscles in several birds and mammals such as the beagle and the jerboa, extra toes in guinea pigs, reemergence of sexual reproduction in the flowering plant hierarchy of mites, webbed feet in adult axolotls, which are like a big salamander, human tails, non-pseudo tails, and supernormally nipples in humans and other primates, color blindness in humans. Atavism is a term in Joseph Schumpeter's explanation of World War I in the 20th century liberal Europe, blah, blah, blah. I'm not really too worried about cultural atavism. Social Darwinism. During the interval between the acceptance of evolution in the mid-1800s and the rise of a modern understanding of genetics in the early 1990s, atavism was used to account for the reappearance in an individual of a trait after several generations of absence, often called a throwback. The idea that atavisms could be made to accumulate by selective breeding or breeding back led to breeds such as the heckback cattle or the heck cattle. This had been bred from ancient land races with selective primitive traits in an attempt of reviving aurochs, an extinct species of wild cattle. The same notions of atavisms were used by social Darwinists who claimed that inferior races displayed atavistic traits and represented more primitive traits than other races. Both atavisms and Ernst Haeckel's recapitulation theory are related to evolutionary progress as development towards a greater complexity and a superior ability. In addition, the concept of atavism as part of an individualistic explanation of the causes of criminal deviance was popularized by Cesare Lombroso. In the 1870s, he attempted to identify physical characteristics common to criminals and labeled those he found as atavistic, throwback traits that determined primitive criminal behavior. His statistical evidence and closely related idea of eugenics have long since been abandoned by the scientific community. Yeah, they've been abandoned. Yeah, because the scientific community doesn't care to accept, the facts being the facts. But the concept that physical traits may affect the likelihood of criminal or unethical behavior in a person still has some scientific support. No duh. So what's the wild type? The concept of that's a wild type banana. If you ever watch, I'm gonna show that because that's hilarious. On Kirk Cameron's show, he had some creationist on talking about the banana, the atheist's worst nightmare. Okay, the concept of wild type is useful in some experimental organisms, such as fruit flies. Okay, so what is a wild type? The wild type is the phenotype of a typical form of a species as it occurs in nature. Originally, the wild type was conceptualized as a product of standard normal allia at the locus, in contrast to that produced by a non-standard mutant alii. Mutant allele can vary to a great extent and even become the wild type if the genetic shift occurs within the population. Okay. So what is epigenetics? Your genes play an important role in your health, but so do your behaviors and your environment. This is what people have to understand. Your choices matter. Okay? Many years ago, when I was a young adult, I was a genetic determinist. I believed in genetic determinism. I was a hardcore supporter of social Darwinism, a hardcore supporter of eugenics. And then as I got older and I started realizing, choices really matter. Choices matter a lot in the development of the phenotype and in the development of health and in the expression of genes. And I think we've all known this. Most in the scientific community knew this. It's not unique to Darwinism. Darwinism recognized this in the beginning, that environment plays a crucial role. Hence the word natural selection. It plays a crucial role in the expression of genes. So epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Unlike genetic changes, epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reads the DNA sequence. Gene expression refers to how often or when proteins are created from the instructions within your genes, while genetic changes can occur which alter which protein is made. Epigenetic changes affect gene expression to turn genes on and off. Since your environment and behaviors such as diet and exercise can result in epigenetic changes, it is easy to see the connection between your genes and your behaviors. How does epigenetics work? Epigenetic changes affect gene expression in different ways. DNA methylation. DNA methylation works by adding a chemical group to DNA. Typically, this group is added to a specific place on the DNA where it blocks the proteins that attach to DNA to read the gene. This chemical group can be removed through a process called demethylation. Typically, methylation turns genes off and demethylation turns genes on. Histone modification. DNA wraps around proteins called histones. When histones are tightly packed together, proteins that read the gene cannot access the DNA as easily, so the gene is turned off. When histones are loosely packed, more DNA is exposed or not wrapped around a histone and can be accessed by proteins that read the gene, so the gene is turned on. Chemical groups can be added or removed from histones to make the histones more tightly or loosely packed, turning genes off or on. Non-coding RNA. Your DNA is used as instructions for making coding and non-coding RNA. Coding RNA is used to make proteins. Non-coding RNA helps control gene expression by attaching to coding RNA along with certain proteins to break down the coding RNA so that it cannot be used to make proteins. Non-coding RNA may also recruit proteins to modify histones and to turn genes on or off. How can your epigenetics change? 1. Epigenetics and development. Epigenetic changes begin before you are born. All of your cells have the same genes, but look and act differently. Now, Maria Konovalenko does a wonderful job of explaining this. As you grow and develop, epigenetics helps determine which function a cell will have. For example, whether it will become a heart cell, or a nerve cell, or a skin cell. Example, nerve cell versus muscle cell. Your muscle cells and nerve cells have the same DNA, but work differently. A nerve cell transports information to other cells in your body. A muscle cell has a structure that aids in your body's ability to move. Epigenetics allows the muscle cell to turn on genes to make proteins important for its job and turn off genes important for the nerve cell's job. Epigenetics and age. Your epigenetics, and this is why everybody needs to read True Age by Morgan Levine. Everybody needs to read Dr. Morgan Levine's book, True Age. Epigenetics and age. Your epigenetics change throughout your life. Your epigenetics at birth is not the same as your epigenetics during childhood or adulthood. Example studies, newborn versus 26-year-old. DNA methylation at millions of sites were measured in a newborn, 26-year-old, and 103-year-old. The levels of DNA methylation decreases with age. A newborn has the highest DNA methylation. The 103-year-old had the lowest DNA methylation, and the 26-year-old had a DNA methylation between the newborn and the 103-year-old. Epigenetics and reversibility. Not all epigenetic changes are permanent. Some epigenetic changes can be added or removed in response to changes in the environment. Example, smokers versus non-smokers. Smoking can result in epigenetic changes, for example, at certain parts of the AHRR gene. Smokers tend to have less DNA methylation than non-smokers. The difference is greater for heavy smokers and long-term smokers. After quitting smoking, former smokers can begin to have increased DNA methylation at this gene. Eventually, they can reach levels similar to those of non-smokers. In some cases, this can happen in under a year, but the length of time depends on how long and how much someone smoked before quitting. Infections. Germs can change your epigenetics to weaken your immune system. This helps the germ survive. Examples, Mycobacterium tuberculosis. Tuberculosis causes tuberculosis, blah, 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 infections. These germs can cause changes in histones in some of your immune cells that result in turning off the IL IL-12B gene. Turning off the IL-12B gene weakens your immune system and improves the survival of the Mycobacterium tuberculosis. Cancer, certain mutations make you more likely to develop cancer. Likewise, your epigenetic changes increase your cancer risk. For example, having a mutation in the BRCA1 gene that prevents it from working properly makes you more likely to get breast and other cancers. Similarly, increased DNA methylation that results in decreased BRCA1 gene expression raises your risk for breast cancer and other cancers. A colorectal cancer being used as an example. Colorectal cancers have abnormal methylation at DNA regions near certain genes, which affects gene expression. Some commercial colorectal cancer screening tests use stool samples to look for abnormal DNA methylation. Nutrition during pregnancy. Okay, a pregnant woman's environment and behavior during pregnancy, such as whether she eats healthy food, can change the baby's epigenetics. Some of these changes can remain for decades. Example, Dutch hunger winter famine. People whose mothers were pregnant with them during the famine were more likely to develop certain diseases such as heart disease, schizophrenia, and type 2 diabetes. Years after the famine, researchers looked at the methylation levels in people whose mothers were pregnant with them during the famine. These people had increased methylation, meaning they were older at some genes, and decreased methylation at other genes compared with their siblings. So some genes were being expressed more, some genes were being expressed less. Or some, actually it would be some are being expressed less, some are being expressed more. So I'm going to move on here. So who's Bruce Lipton? Okay, Bruce Lipton is an American developmental biologist noted for his views on epigenetics. In his book, Biology of Belief, he claims that beliefs control human biology rather than DNA and inheritance. Lipton's contentious claims have not received attention from mainstream science. I don't believe that beliefs control human biology rather than DNA and inheritance. I think they kind of work together. Okay. Biography, blah, blah, blah. Lipton received his BA in biology in 1966. That's his education, reception, books. Okay. Who's Dr. Joel Wallach? Wallach is a veterinarian, naturopathic physician, author, and lecturer who played a major role in the development of the market of liquid vitamin mineral supplements. He became nationally known through his widely distributed audio tape, Dead Doctors Don't Lie. Periodically, critics question his background and some of his views. The present account specifically analyzes some of the objections raised in the third edition of the Medical Resource Manual. Joel Wallach was born in West St. Louis County on Okay, so Joel Wallach's background and profession. During the early 1960s, environmental pollution and other ecological factors were thought to cause the premature death of captive animals and possibly of humans. The National Institutes of Health awarded the St. Louis Zoological Gardens a large grant to identify these factors. The project required a well-rounded wildlife veterinarian and pathologist. Wallach was hired for this position, which provided him with the opportunity to autopsy a wide variety of captive wild animals dying of natural causes in zoos at St. Louis, Chicago, Los Angeles, Jacksonville, Memphis, Atlanta, Georgia. Wallach conducted comparative autoptic studies on primates as well as on humans. His work was progressing until 1979 when he discovered pancreatic lesions in the offspring of a family of inadequately fed rhesus monkeys, which histologically were identical to those observed in patients with cystic fibrosis. The finding led Wallach to propose that cystic fibrosis in humans was also caused by nutritional imbalances and or deficiencies, as cystic fibrosis is considered to be a genetic disorder. Now, this has to do with epigenetics and nutrition, okay? His views seemed so extreme at the time that he was dismissed from his position at Yerkes Primate Research Center. Scientific evidence would subsequently catch up with his bold hypothesis. But in the interim, with little or no chance of finding an appropriate position in his profession, Wallach decided to change careers. In 1980, Wallach joined the faculty of the National College of Naturopathic Medicine in Portland, Oregon, where he taught in the area of nutrition while pursuing his naturopathic doctorate degree. Okay, in 1982, after obtaining his naturopathic doctorate degree and license in 1982, Wallach went into private practice in Cannon Beach, Oregon, specializing in the nutrition treatment of cystic fibrosis patients. In 1987, to continue his research on cystic fibrosis, he traveled to China with his new wife, Dr. Ma Lan, a Chinese physician, to conduct a study of Harbin Medical University, or at Harbin Medical University from 1990 to 1993. Wallach worked legally as a naturopathic physician for the Hospital of Santa Fe Monica in Tijuana, Mexico. Dr. Wallach's publications, I'm not going to go over that, but you should, let me go. A partial list of Dr. Wallach's publications. He's written many, many books. He's authored and co-authored during the period of 1965 to 1994, comprises 55 titles. 20 of his papers were published in the Journal of American Veterinary Medical Association, JAVMA. 6 reviews appeared in professional books, the remainder in other professional veterinary journals. Wallach, furthermore, is the co-author with W.J. Bouvier of an authoritative treatise, Disease of Exotic Animals, Medical and Surgical Management, published by W.B. Saunders and Company, Philadelphia, 1983. He also authored with Ma Lan 2 books directed to lay audiences, Let's Play Doctor and Rare Earths, Forbidden Cures, both published by a Double Happiness Publishing Company, Bonita, California. His first paper, appearing in 1965, describes goiterogenic hypothyroidism in feeder lambs. Subsequent articles and reviews deal blah blah with common diseases, treatments, birds, blah blah blah. So this guy knows what he's talking about. That's what you need to understand. When it comes to medicine and nutrition, he knows. Genetic and epigenetic contributions to human nutrition and health, managing genome-diet interactions. Okay, so let's take a look through here. So this is the abstract, and I'm going to read this quickly. The Institute of Medicine recently convened a workshop to review the state of various domains of nutritional genomics research and policy, and to provide guidance for further development and translation of this knowledge into nutritional practice and policy. This is what's known as nutrigenomics or nutritional genomics. It's a whole science now called nutrigenomics. Holds the promise to revolutionize both clinical and public health nutrition practice and facilitate the establishment of a genome-informed nutrient and food-based dietary guidelines for disease prevention and healthful aging, individualized medical nutrition therapy for disease management, and better targeted public health nutrition interventions that maximize the benefit and minimize the adverse outcomes within genetically diverse human populations. Okay, keep that in mind. Genetically diverse human populations. Humans are genetically diverse. Keep that in mind. Yes, minor differences. Minor differences make big differences in how it's expressed in the phenotype, which is why epigenetics is so important. As the field of nutritional genomics matures, which will include filling fundamental gaps in nutrition of nutrient-genome interactions in health and disease, and demonstrating the potential benefits of customizing nutrition, prescriptions based on genetics registered dietitians will be faced with the opportunity of making genetically driven dietary recommendations aimed at improving human health. Public health nutrition continues to be challenged by increasing expectations from the food supply on one hand, and fundamental gaps in nutrition knowledge on the other, which can constrain the development and implementation of nutrition and food policy. Number 1, current demands on the food supply are no longer limited to ensuring general safety and preventing micronutrient deficiencies. Increasingly, there is interest in engineering medical medicinal qualities into the food supply to enable diets that promote health and nurture a sense of well-being that transcends the mere absence of disease by improving biological functions and even increasing lifespan. Okay, unquestionably, nutrition is one of the primary environmental exposures that determines health. Common human chronic diseases, including type 2 diabetes, metabolic syndrome, cardiovascular disease, and neurological disease, and many cancers are initiated and or accelerated by nutrient/food exposures. However, it is also recognized that chronic diseases are complex in their etiology and include a substantial genetic component. This is where epigenetics comes in. So nutrigenomics or nutritional genomics will, while centered on the biology of individuals, distinguishes itself from other omics fields by its unique focus on disease prevention and healthy aging through the manipulation of gene-diet interactions. Nutritional genomics promises to revolutionize both clinical and public health nutrition practice and facilitate the establishment of a genome-informed nutrient and food-based dietary guidelines for disease prevention and healthful aging, individualized medical nutrition therapy for disease management, and better targeted public health nutrition interventions, including micronutrient fortification and supplementation that maximize, now remember that, micronutrient fortification and supplementation that maximize benefit and minimize genetically diverse human minimize adverse effects in genetically diverse human populations. In 2006, the Institute of Medicine convened a workshop, blah, blah, blah. So let's get down into here. So, origin of gene-nutrient interactions. The human genome and genetic variation present within human populations is in part a product of adaptive evolution to an often scant and unpredictable food supply. Single nucleotide polymorphisms, which are common single base pair differentiations in DNA sequence, represent a primary form of human genetic variation of the approximately 10 million SNPs in the human. Indeed, many. Let's go. This is not really important to know for our purposes. Folate metabolism, a conduit between the nutrient environment and the genome. This is one of the reasons I take folic acid. This is another reason why certain people that have issues with methylation should probably take methylfolate. And remember, I'm not a doctor, I'm not a chiropractor, I'm not a nutritionist, I'm not a dietitian, I'm not a nurse practitioner, I'm not an LPN, I'm nothing like that. Don't take this video as health advice. Folate is a B vitamin that functions as a metabolic cofactor by carrying and chemically activating single carbons for the biosynthesis of purine nucleotides and thymidylate, and for the remethylation of homocysteine to methionine, and metabolic network known as folate-mediated one-carbon metabolism. Okay, methionine in turn can be adenosylated to form S-adenosylmethionine, or SAM, which is a co-substrate for numerous cellular methylation reactions. Purines and thymidylate are required. Now remember, as you get older, you get less methylation. Therefore, what might folate do for you? The human genome is methylated on cytosine bases present within DNA at specific CpG sequences. Lysine residues on histone proteins bound to DNA are also targets of SAM-dependent methylation. Methylation of chromatin, a complex of DNA and histones, regulates the expression of numerous genes throughout the genome. So you can see folate plays an important role in gene expression. Methylation is associated with inactivation of an X chromosome in females and silencing genetically imprinted genes where only the maternal or paternal allele is expressed and the other is silenced by methylation. Methionine and thymidylate biosynthesis are the most sensitive pathways within the folate metabolic network, and their impairments compromise the rate and fidelity of DNA synthesis. So keep that in mind. Folate is necessary so that your DNA says what it's supposed to say. That's what fidelity means, okay? The folate-dependent biosynthesis of nucleotide precursors for DNA synthesis and of the SAM for genome methylation is dependent on the availability of many vitamins, including B12, and this is why you should take more than just folate. B12, B6, niacin, riboflavin, and minerals, zinc, and cobalt, and is subject to regulation by other nutrients not directly involved in DNA or cell biosynthesis, iron, vitamin A. Therefore, folate-mediated one-carbon metabolism is a conduit that enables communication between the cellular nutrient environment and regulation of genome impairments in one-carbon metabolism and the SAM cycle, in particular induced by nutritional deficiencies. Okay, so so I'm going to move on. Now, when I go a little further, I'm going to show how to release the wild type through nutrition. I'm going to do a series of these, I think. But this is 5 ways to optimize genetic potential through nutrition. Now, keep in mind, Dr. Wallach was a veterinarian before he was a naturopathic physician. And it's important to understand, why is it that veterinary science is so beyond medical science? You know what I mean? It's ridiculous. Or human medical science, I should say. So, number 1, take time. Okay, so 5 ways to optimize genetic potential through nutrition. Take time to understand the big picture, step back and think about all you are trying to accomplish. If you are working on genetic improvement within your herd, it is not a good idea to short your cows during gestation. The cow is the only source of nutrients for the calf. Okay? If you aren't meeting the cow's requirements during gestation, you aren't meeting the fetus's requirements either. That's also true for humans. Keep that in mind. This is why gestational nutrition is so important for women, and as well as you know what I mean? It's very important that women stay very well nourished. Optimize health through nutrition. Cattle require nutrients to support normal growth and development. Many times we focus on vitamin and mineral nutrition, but other important nutrients like protein and energy are also necessary for cattle to reach their genetic potential. They're also important for people. When I started doing the starch solution by Dr. McDougall, it didn't agree with me. The high starch, low protein diet did not agree with me. Protein and energy are not only needed by the cow, but are also required to help rumen microbes function, replicate, and thrive. Well-fed rumen microbes provide energy and high-quality protein to the animal, supporting growth and development. Now, we don't have a rumen, but we do have a microbiome. Okay. In addition to supporting growth, protein provided to the cow allows maximum forage digestion and utilization by the rumen microbes. Highlighting protein and energy needs doesn't mean we should minimize the importance of a good vitamin and mineral program. Vitamins and trace minerals are critical for health and immune function, and sick cattle rarely meet their genetic potential. Number 3. Know what your cattle are eating. It's hard to know whether you need to supplement protein or energy if you don't know what you are starting with. Okay, and this is true with us. This is why when I go further, I'm going to talk about to quote Frank Tufano, the importance of an ancestral diet, going back to an unrefined diet, not like, you know, some people go crazy with it, but looking at what our great ape cousins eat, and what hunter-gatherer groups eat, and what people that live a really long time, like the Okinawans, eat, and looking at that and making dietary adjustments based on that. In comparison, we don't generally buy a bull without knowing his expected progeny difference numbers. So why do we feed cattle without knowing the nutrients in the feed? Keep that in mind. Why do you eat food without knowing the nutrients in your food? Whether you're feeding cattle or have cows grazing, understanding nutrient content and feed intake will help identify the gaps in what the cattle are consuming. Okay? Develop a nutrition plan. Okay, and I'm going to talk about that. Ask for help. Okay, this is when you should go to a nutritionist, a doctor, somebody like that to help you with your nutrition. Genetics plus environment equals phenotype. Genetics plus environment equals phenotype. When I move forward, I'm going to go over the biopsychosocial model And I'm also going to go over the traditional diets, and that will be in a later video, and that's all for this video.